Welcome back to MLT 240. Today in lecture we're going to cover trematodes or flukes. Trematodes fall in the class Digenea and they're commonly called flukes because they belong to the phylum Planiathehelminth along with our cestodes. Flukes are non-segmented, they're dorsally flat, they vary in length from a few millimeters to several centimeters. They have simple digestive tracts with no anal opening, and their waste products are regurgitated. There's two types of trematodes present in humans, those that live in the intestines or other organs such as the liver and lungs, and that are hermaphroditic. And there are those that live within the abdominal blood vessels of our host. These are known as our blood flukes, and they are unisexual. A lot of our flukes have operculated eggs, meaning they look like they have little lids on them, except for our schistosome species. And most of our species of trematodes are hermaphroditic, except for our blood flukes or our schistosomes. Adult flukes have two cup-shaped muscular suckers called the acetibula. They have an oral sucker and a ventral sucker. Here is the general life cycle of our trematode. The trematodes really have a complex life cycle in which a specific freshwater snail is required as an intermediate host for each species of fluke. The larval stage emerges from the egg in fresh water and enters the snail, where the larva undergoes several cycles of asexual multiplication. The final larval stage leaving the snail is known as the cercaria. Many hundreds of cercaria result from asexual multiplication of each mercidium that enters the snail host. Motile cercaria of the blood flukes then enter the skin of the human when contact occurs in infected freshwater. So let's look over here at the diagram. Eggs pass through feces, so they're in the water. Eggs hatch and liberate the larval stage mercidium, and then that stage of the parasite are able to develop in a certain type of snail. So you have to have a type or a species of snail involved. From there, the larval stage can leave the snail, and it's known as a cercaria, and the cercaria can enter the human host when the host is in fresh water. There's three species of blood flukes or trematodes that we're going to cover. There's Schistosoma mansoni, which is the Manson blood fluke. There's Schistosoma japonicum, which is the blood fluke. And there's Schistosoma hematobium, which is the bladder fluke. So pathology facts about schistosoma. Schistosomiasis ranks second behind malaria as a cause of serious worldwide morbidity and mortality. It mainly affects children, but it can also affect adults in epidemic areas. Initial reaction may be dermatitis from cercarial penetration causing swimmer's itch. Acute phase of the first heavy infection presents with symptoms similar to typhoid fever-like symptoms. You may end up with a cough, fever, myalgia, malaise, hepatosplenomegaly, which is an enlargement of the liver and spleen. Complications can occur due to the granulomas that form in the eggs or around the eggs in the liver, bladder, the central nervous system, and other tissues. Granulomas is referring to a mass or inflamed tissue. You may have cirrhosis of the liver, bloody diarrhea, bowel obstruction, hypertension, or toxic reactions with schistosoma. Here's our schistosoma life cycle again, and I just want to reiterate the cercaria is going to be entering the skin, but our three blood fluke species that we're talking about are Schistosoma mansoni, japonicum, and hematobium. They have to have that intermediate host of a species of snail, okay, in order for schistosoma or our flukes to complete their life cycle. Here is an image just showing you some of the different stages of life or development of our schistosomas. You can have the egg, which can be found in feces or urine samples of our schistosomes. There's the mycelium stage. There's the sporocyst stage that usually is completed within the snail during the life cycle. The saccharium, this is the larval stage that actually goes through the skin and penetrates the skin for humans to get it. And these are adult worms. For our lab ID, eggs can be present in the stool in infections with all our schistosome species. Stool examination should be performed when infection of Schistosoma mansoni or Schistosoma japonicum are expected. Also, you can do urine examinations if you suspect Schistosoma hematobium and look for eggs. It's recommended for Schistosoma hematobium that collection be done between noon and 3 p.m. You can also do urine examination for Schistosoma japonicum. Another way to ID is to do tissue biopsy if you're not able to find eggs in stool or urine examinations. Tissue biopsies of the rectal area for all species may yield eggs. You could also do a tissue biopsy of the bladder if you suspect Schistosoma hematobium. Now let's look at images for the eggs for each of our blood fluke species. Schistosoma hematobium. The egg species are large and have prominent terminal spines at the posterior end. So here is the example of the eggs, and here is the terminal spine at the posterior end. Here's the terminal spine. For Schistosoma mansoni, these are large eggs and they have a characteristic kind of oval shape with a prominent lateral spine near the posterior end. The anterior end is tapered to lightly curved. So here is that prominent lateral spine. You can see that but in both these images of eggs. The anterior end is tapered to slightly curved. So anterior end would be this end here, because the lateral spine is closer to the posterior end. This would be the posterior end with the lateral spine. This would be the anterior end, tapered to slightly curved. This is showing you under the scope what a Sacchariae would look like. This is what would actually penetrate the skin of the human and how you would end up infected with a schistosome or a blood fluke. For Schistosoma japonicum, the eggs are typically oval to subspherical, so they're pretty round. They are very small or have a very small lateral spine. And overall, this species is smaller than the other species of schistosomes. And here is that very small little lateral spine. And there is a 10 micron scale, so you can see the size of Schistosoma japonicum. And this is Schistosoma japonicum's Sacchariae. Again, this would be what enters or penetrates the skin of a human. As far as distribution for our schistosome species, Mansoni is found in South and Central America and the Caribbean. Schistosoma hematobium is found in Africa and the Middle East. Schistosoma japonicum is in the Far East. Now let's talk about the other trematodes, our hermaphroditic trematodes. These are either going to be found in the intestinal tract or other tissues. We're going to talk about Fasciolopsis busky, which is the large intestinal fluke, Fasciola hepatica, which is the sheep liver fluke, Clonorchus sinensis, which is our Chinese liver fluke, and Pergonimus westermanni, which is the oriental lung fluke. Fasciolopsis busky is known as our large intestinal fluke. The transmission is ingestion of encysted Metasacchariae in raw vegetation. This parasite is also very common in pigs. In humans, the maturation of this species takes approximately 3 to 4 months, and adults live in the small intestine. Symptoms of this parasite include bowel mucosal ulcers, hypersecretion, and occasional hemorrhaging around the worm attachment site. Heavier infections can cause pain, nausea, mucoid diarrhea, anemia, intestinal obstruction, malabsorption, edema, and eosinophilia. Even death can occur with very heavy infections. The life cycle is very similar to our blood flukes in that we have to have a snail as an intermediate host, but instead of the Cercariae penetrating the skin, we get the Metacariaceae off of vegetables that we actually ingest. And of course, the pig are also a common host for this parasite. For the lab ID of F. busci and the distribution, you can recover eggs in fecal samples, but the eggs of F. busci and F. hepatica are too similar to differentiate, so you're not going to be able to tell which species is which. Species diagnosis is going to depend on clinical symptoms, travel history, and the recovery of the adult F. busci, or all three factors. F. busci is prevalent in China, Vietnam, Thailand, Indonesia, Malaysia, and the Indian subcontinent. Here is an image of the egg and the actual adult fluke for F. busci. The egg is oval. It's large. The size is anywhere from 90 to 150 microns. If you look over to the adult fluke, you can see we're looking at the dorsal side, and all these darker brown lines are representing the uterine branches that are holding eggs inside. Remember, these are hermaphroditic, very similar to our tapeworms. They have both sex organs. Next up is Fasciola hepatica. This is also known as the sheep liver fluke, and it causes sheep liver rot. Humans can become accidental hosts by ingesting encysted Metacariae on raw vegetation. In humans, the maturation to adults or adult flukes is approximately 3 to 4 months in this species. The approximate size for an adult fluke is somewhere between 30 millimeters by 13 millimeters. The natural definitive host for this species are sheep. So, pathology and symptoms for Fasciola hepatica include mechanical irritation, toxic worm metabolites, and mechanical obstruction. This is from migrating larvae, cause local irritation. With this species, you can also have fever, hepatomegalia, and eosinophilia. Large flukes in the bile duct induce portal cirrhosis and heavy infections. Jaundice can occur, bile duct obstruction, diarrhea, and anemia. Other symptoms can include pruritus or itching, urticaria, and cough. Here's our life cycle for Fasciola hepatica. Again, humans are accidental hosts because we ingest Metacariae on some type of vegetation. But again, there has to be some kind of snail or species of snail involved for the life cycle to complete for Fasciola hepatica. For lab ID and distribution for F. hepatica, we can use microscopic identification of eggs. It is usually helpful in chronic cases. Eggs can be recovered from stool samples or other samples such as duodenal drainage or biliary drainage. Remember, F. hepatica is morphologically indistinguishable from F. busci as far as the eggs. We can use antibody detection tests. These are especially useful in early invasive stages of the parasite when the eggs are not yet apparent in stool samples. Human infections with F. hepatica are usually found in areas where sheep and cattle are raised and where humans consume raw watercress. And this is because watercress are raised close to bodies of water, and that's where the vegetation becomes contaminated with the parasite. Areas include Europe and the Middle East and Asia. We're looking under the scope in Lab ID for F. hepatica. These eggs are going to look very similar to F. busci. The eggs are going to be oval or elliptical in shape, and they may have a little operculum or a little lid that you may or may not see it. These eggs are going to have a thin shell, and they may have a slightly thicker appearance at the abopercular end, meaning the end opposite of the operculum. The size is again going to be about 90 to 150 microns, so the same size as F. busci. Here is an image of Fasciola hepatica, the adult fluke. This is the anterior end, and you can see those uterine branches. You can't see it as well as the other fluke that we looked at a few minutes ago. Approximate size is 30 millimeters by 13 millimeters for the adult. The next species of trematodes is Clonorchus sinensis. It's known as the Oriental or the Chinese liver fluke. The transmission is ingestion of encysted Metaceriae in undercooked fish. Light infections are common and may be asymptomatic. In the acute phase, you may have abdominal pain, nausea, diarrhea, and eosinophilia may appear. In long-standing infections, you may have long-term impairment of the liver. Maturation of this species takes approximately 1 month. Adult flukes can measure anywhere from 10 to 25 millimeters by 3 to 5 millimeters. They can reside in the small and medium-sized biliary ducts. In addition to humans, carnivorous animals can serve as a reservoir and as host. Here is our life cycle for Clonorchus sinensis. Again, we become infected by eating undercooked fish, and we get the Metaceriae from that undercooked fish. And again, here is our snail that has to be present as our intermediate host to complete the life cycle for our trematodes. For ID, we can have microscopic demonstration of eggs in stool or duodenal aspirate. Eggs for this species are usually passed intermittently, so sequential stool examinations may be necessary. The eggs are percolated, and they have a light bulb shape with prominent shoulders, and we'll look at what that looks like here on the slide that's coming up. The adult fluke can be recovered by surgery. We can also do the ELISA test to test for this particular parasite, and Clonorchus sinensis is found in the Far East, especially in southern China. For Clonorchus sinensis, these are small operculated eggs. So here is the little lid, the operculated lid. The size is only 16 to 30 microns. The operculum is at the smaller end of the egg, and it's going to rest on the end where there's visible shoulders. So these are the shoulders, okay, or what's considered the shoulders. On the opposite end, the larger end, there's going to be a small knob that is visible. And sometimes the mycelium is visible inside the egg. A lot of times students say this looks like a little mouse or looking down on a little mouse. So this would be the head, and the shoulders kind of look like the ears, and the little knob looks like the tail of the mouse. The last fluke that we need to discuss is Paragonymous westermanni. It's known as the Oriental lung fluke. The transmission is ingestion of incited Metaceriae in undercooked crab or crayfish. Symptoms include chronic chest pain, cough, blood-tangled sputum, lung infiltration, and nodules, and even abscesses. Adults present with fibrous cyst. Eggs pass through cyst and rupture into the bronchioles, causing a cough. In chest radiographs, you may have findings and symptoms that resemble tuberculosis. Other developments for this parasite can include chronic bronchitis and increased fibrosis. Here is our life cycle for Paragonymous westermani. Again, we're going to come infected with this parasite when we eat undercooked crayfish. And just like our other flukes that we've been talking about, we have to have that particular species of snail that matches up with this parasite in order for the parasite to continue its life cycle. For our lab ID for Paragonymous westermani, the diagnosis is demonstration of eggs in stool or eggs in bloody sputum. They might not present until 2 to 3 months after infection. The average size of eggs is 85 microns by 55 microns. The eggs usually present a yellow-brown color, ovoid or elongated with a thick shell. They're often asymmetrical with one end slightly flattened. At the large end, the operculum is clearly visible, and the opposite end is thickened. Paragonymous westermani is usually found in the Far East and also parts of Africa and South America. This is what Paragonymous westermani would look like for our lab ID under the scope. You have the operculum over on this end of the lid, and you have that thickened shell over on the opposite end. This concludes our trematode lecture. If you have any questions, please don't hesitate to reach out.